next talk will be from Alex Libby, who will be telling us about dynamic and sustained representations in working memory.
Hi, my name's Alex Libby, and I work with Tim Buschman, and I'd like to tell you about how sustained and dynamic representations reduce interference in short-term memory. So predictions are key to cognitive processing, and this is because the events in our lives occur in sequence, and we can learn the transition probabilities between these events, and that allows us to optimize our sensory processing. And so, for example, if you're watching a tennis game, you can't understand any given moment without remembering the history of events that took place. So what we're addressing with this study is how does the brain preserve short-term memory in these sequences of events? To study this, we designed an implicit sequence learning paradigm in which mice passively listen to sequences of sounds. And these sequences begin with one of two contexts. For context AB, A is always followed by B and frequently followed by C, and all sequences end in D. And expected trials like these occur about 2/3 of the time. On rare trials, AB is followed by C prime, and these unexpected trials occur 20% of the time. The remaining 12% of trials were ambiguous stimuli, and I can talk about those offline. To balance our context, we have another one, XY, in which XY predicts C prime this time and is rarely followed by C. So what we have is a context, A, B, or XY, followed by a predicted stimuli, C or C prime. And so our expected trials are A, B, C, D, XY, C prime D, and our unexpected trials are A, B, C prime D, and XY, C, D. So what's important to remember about this paradigm is that it's balanced, in that both contexts occur equally per day, as do the predicted stimuli, so the mouse can't make a prediction before any given sequence begins. To understand how these statistical regularities affect neural activity, we chronically recorded from auditory cortex in head-fixed mice. On day 1, the mice were completely naive to the sequences, and they listened to about 1,000 trials per day. And by day 4, they were very well experienced with these regularities. So let's take a look at an example cell, and on the x-axis is time, and on the y-axis is firing rate, and so you could see how it responds to different elements of the sequence. And this particular cell has a preference to context AB, which are shown in the warm colors, pink and orange. But we want to understand how the entire population encodes sensory inputs. And so in order to do that, we can divide our trials by whether it was an A or an X trial, and look at the firing rate over time. And so first we begin by looking at the initial part of the sequence, the A or X period, and we take the average firing rate and just train a linear SVM to differentiate between A and X trials. And I should say, on all our classifiers, we balance for expectation. And so what we can do is we can take our ND firing rate activity and project it onto a 1D sensory axis, in which positive now indicates X encoding and negative indicates A encoding, and we can look at how the population activity evolves over time. And so all I want you to see here is that there's an initial separation of the A and X context that diminishes over time. So we can do the same thing for our predicted stimuli, train a classifier to differentiate between the C and the C prime trials, taking the firing rate activity during the C and C prime stimulus. And to understand the relationship between context and predicted stimuli, I'm going to project the neural activity into a 2D state space, in which the y-axis now is our predicted stimuli encoding, and the x-axis is our context. So let's take a look at day 1 at the initial part of the sequence, so either A or X was played. So we have this sort of 4-quadrant encoding space. And if I take trial type XYC prime D, you'll see that it moves to the right because it's correctly encoding X. Likewise, ABCD moves to the left because it's correctly encoding A. And the same is true for our other 2 trial types. The question is, what happens after learning? So we can take the same plot on day 4. And what you'll see is if you look at the X trials, which are the green and the blue plots, you'll see that, again, they move right, but they also move upwards to encode for C prime. And this is before the predicted stimuli is even played. So as you can see, there's sort of a prediction based on this encoding for the stimuli before it even comes on. And so to understand where this prediction is coming from, we look at the relationship between these 2 axes by measuring the angle between them. And we find that on day 1, it starts out being orthogonal and that this decreases over time. And so as the axes become aligned, activity along one axis induces activity along the other. And this is great because it initially gives us this prediction at the beginning of the sequence, but it causes a problem by the C/C' period of the sequence. So now I'm going to plot activity dynamically, and I would like you to focus on the pink, which is the unexpected condition ABC'D. And you could see it initially correctly encodes for A, but as C' is played, it shoots across and begins to encode XC'. And this is because as C' is played, we're inducing activity along our other axis and beginning to encode X. And so what we're calling this is a post-diction, or an altering of your memory of the past to fit the present. So this begs the question, is there no memory of the past during the C/C' period? So just one more classifier that I trained to differentiate between A, B, and XY, but this time using the firing rate activity during the C/C' period. So I'm going to plot the exact same state space as before, but this time I'm replacing the x-axis with this new context memory encoding axis. And what we see is if we look at encoding during that C period is that all 4 trial types are in their correct quadrants, most notably those unexpected conditions, which are in pink and green. So we have these 2 sensory axes, or these 2 axes for context, the sensory and the memory. And so the A sensory is aligned with C sensory, which gives us that post-diction and prediction. Meanwhile, our AX memory axis is orthogonal to C sensory, and that keeps the memory intact. And so how does that happen? Well, it could be just through independence. You may have some set of cells during this initial AX period that are selective to A or X, and another set of cells sort of becomes active or selective during the latter half of the sequence, and then that gives you this orthogonalization. Alternatively, you could imagine that all of the cells are selective initially, and that they sort of rotate their selectivity over time to be orthogonal to that original sensory axis, thereby maintaining the memory of the past. And so we wanted to understand what is the temporal selectivity profile of our cells. So again, we can divide our trials by X in purple and A in purple and X in green, and we can calculate a z-scored firing rate difference over the course of the sequence. And so we can do this for all of our cells. And we take these temporal selectivity profiles and we cluster them using an unsupervised technique called the phenograph. And we got 4 clusters out of this, namely 2 functional clusters. One are stable neurons that sustain their selectivity to contextual sounds over time, and switching neurons that switch their initial selectivity after the onset of the AX period. So how do these 2 cell types relate to the sensory and memory axes? To do that, we can group the contributions of these cell types to the classifiers, and I'm going to just take the ones that have initial negative selectivity and flip their activity upwards. So on the y-axis, now if it's positive, that means its preference matches that cell's preference during the AX period. So if we look at the asensory classifier, it's going to have positive weights for both those stable and switching neurons, and that just means if we looked at the firing rate of those neurons, they fire more for their preferred stimuli with respect to that AX period, and that gives us our initial sensory axis. And we do this so we can compare to our context memory axis, and now we see those stable neurons also maintain their preference relative to the AX period, but the switching neurons switch their selectivity. And so what this gives us is the rotation, because the preference for the stable remain the same, whereas the switching become negative, and this gives us an orthogonal memory axis. And you can imagine if the sensory axis were built from stable neurons only, you wouldn't be able to do any type of rotation, and if it was all switching neurons, you would rotate 180 degrees, and again, you wouldn't have an orthogonal axis. So both are really needed for this rotation. In conclusion, predicted sequences align sensory representation, but this alignment causes an overwrite of our AX sensory information. But still, the context information is preserved along an orthogonal axis. And the cell types that build that orthogonal axis are both switching and stable cells. And so what we think is that you really need sustained and dynamic representations for short-term memory. I'd like to thank you for your attention, and I also want to again thank the Buschman Lab for all the feedback and support.
We can take some questions if Sonia's coming up.
Very nice. Maybe I missed it. So do you have any indication that the animals actually learn the sequence, so that they're expecting the specific stimuli? Do you get, for example, stronger responses to if the stimulus is unpredicted? Maybe related to that, maybe I misunderstood it, but it looked to me that the switching neurons actually switch long before the unexpected stimulus is coming. Maybe I misunderstood.
Okay, so I think there's two questions. The first one is we have, it's just passive listening. So our way of telling like whether or not the experience is changing how the mouse thinks, I guess, is only by looking at the neural activity. We don't yet have a behavior to track that. In terms of your second question, can you repeat your second question?
So the last plot you showed, the switching neurons, it seemed they were switching before the unexpected stimulus was shown. They went to the other space. So that maybe the animals, it's learning both sequences, and some mode in the network is tracking the less likely one or something like that?
Right, so we looked to see if each of the individual clusters referred to like the 4 trial types, and that wasn't the case. We also think that the reason the switching neurons are there in place is to maintain the memory, and it's almost like it's there. It wants to switch before it turns on so that it can have that orthogonal space before the C or C prime turns on. And one thing that I've started to track is whether the switching becomes later and later over days, and it seems like it does, but it's still a preliminary analysis. Thank you.
Hi, really cool talk.
Thank you.
I was wondering, because you mentioned that you recorded in both A1 and A2, if you could localize individual units to these different regions, and how does the evolution of stimulus representations over time differ between these two?
Yeah, I really want to look into that. So it's pretty tricky. So I approach the auditory cortex going straight down, so that it's very hard to record from an awake mouse if it has sort of a perpendicular electrode in its brain. But and so differentiating between A1 and A2 might be difficult with the technique that I use, but we are going to look into whether cells identified sort of in higher electrodes have different properties than those that are deeper, which would be in A1. Thank you.
All right. Well, thank you very much, Alex.